Good morning, good afternoon, good evening, depending on where you are. I would like to thank One Lambda and the organizer for giving me the opportunity to talk about our own experience with AllType FASTplex version 2. This is my disclosure slide. I will start my presentation by talking about AllType FASTplex version 1 validation. Then I will talk about the validation of AllType FASTplex version 2. At the end, I will share with you some of the preliminary work that we did in collaboration with One Lambda to validate AllType FASTplex version 2 using the alternative method. In our lab and before implementation of NGS, we used to divide our method of typing into two main categories. For solid organ, we used to do SSO low resolution beads for A, B, C, D, R, SSO high definition for DQ and DP. On the other hand, for bone marrow transplant, we used to do SSO by XR beads for A, B, C, D, R, SSO low resolution for 3, 4, 5, and SSO high definition for DP and DQ. We used to confirm only HLA A and B by high resolution SSP trays. Based on the limitation of the previous method, this is a snapshot of our report. On the top of the report, you can see the high resolution typing, and in the bottom, you can see the non-excluded alleles based on CWD version 2. These that are highlighted in yellow are the alleles that have been added in the CIWD version 3 that was published last year and become mandatory by ASHI and NDP to resolve them as well. With the method that we were using at this time, there was no way to resolve these alleles. That's why we have to move and update our method of HLA typing to NGS. This is just a timeline to give you an idea about how it usually takes for a lab to implement a new method. So we started in September of 2019 and we went live on March 2021, although the actual validation only took us 3 months to finish. During our validation, we used 320 samples, which is equivalent to 640 alleles from our banked DNA, which has been previously typed by high resolution and medium resolution typing. Allele frequency of these samples were calculated and samples were selected to ensure we have at least more than 80% coverage of all common alleles. Out of the 640 alleles that we tested, we found 5 discordant results, 2 in HLA-A, 1 in DR beta 1, and 2 in DP beta 1. If you look here at the discordant result, with our AllType FASTplex, the result for A0201 and the other allele was O224. However, our reference method detected that it's an A01. When we looked further into this issue, we found that it was a sample carryover in SSO, and FASTplex was the right typing. The next discordant was an A0201 3101 null by AllType FASTplex. However, our reference method was not able to detect the null alleles. We used SSP trace, but it was not able to detect the null alleles. When we investigated further and we repeated the typing using an alternative NGS method, we found that it's actually agreeing with the AllType FASTplex, which is 0201 and 3101 null. The third discrepancy was DR beta 101 with AllType FASTplex, but with our reference method was DR beta 01 and 04. And this was a case of allele dropout. When the samples were repeated with using a different DNA, we found that it's actually matching our reference method, which is DR beta 101 and DR beta 04. The last example here was with AllType, it was a homozygous DP beta 1 2301, but with our reference method, it was DP beta 01, DP beta 1 0401 and 2301. When we looked at the SSO string, we found that DP beta 1 0401 and DP beta 1 2301 were in the same string, and the technologists just took the common typing and forgot the other one. So in conclusion of objective 1, AllType FASTplex provided more than 99% concordance with our reference method. Miniseq, which we used as our platform, provided solution for small-size HLA lab that is interested in NGS implementation in a more economical way. As with any method, there are some limitations. One of the limitations of AllType FASTplex version 1 is we didn't sequence exon 1, and as a result, these are the alleles that we cannot exclude using this AllType version 1. All of them are rare alleles except the DR beta 1210, which is part of the DR beta 1201 G or P group. The chemistry of AllType FASTplex used the dual index strategy, I5 and I7 indices. The kit contained a total of 24 unique sample barcode I7 indices and 4 unique FASTplex universal barcode I5 indices to allow up to 96 samples per run. If you are a small to medium-sized lab like our lab that analyze only 24 samples per run, you can pool all these 24 unique barcode samples into one pool. However, if you are a big size lab that utilize over 24 samples per run, then you need to pool the library into either 2, 3, or 4 tubes, depending on the size of your samples. Although these previous two limitations had negligible impact on the quality of the run, One Lambda fixed these in our AllType FASTplex version 2. So FASTplex version 2 comes with a supplementary primer mix for amplifying exon 1 for class 2 gene, DR beta 1, DQ beta 1, and DP beta 1. So all these list of alleles that I mentioned earlier that was not able to be resolved using the AllType version 1 are now being resolved. The other thing also that had been improved in AllType FASTplex version 2 is the redesigned sample and universal barcode reagent. And now you can have only one single tube library processing even for more than 90 or even for up to 96 samples. So whether you are a small lab that utilize only 24 samples, or whether you are a medium or big size lab that utilize 96 samples, you can pool your library into one tube. FASTplex version 2 achieved 100% concordance with version 1. We just noticed when we looked at the total number of reads comparing the FASTplex version 2 to the version 1 that the total number of reads of the ABC locus was very similar between the two protocol, but for DR, DQ, and DP, the total number of reads were significantly higher, and probably this is because of the addition of the exon 1. The length of read were similar between version 2 and version 1, and the same for the depth of read. The average depth of read were between 300 to 1400x. But we also notice in DQ exon 1, the depth of read were more than 5500x. However, the rest of the DQ exon were similar to the others, which is 300 to 1400x. And you can see it in this figure, which shows the uniformity of the reads between the exon. You can see that there was a little bit of uniformity between the exons, but because of the depth of read is 5000 in exon 1 compared to 300 to 1400 in the other exon, you still have sufficient read to call the right alleles. So conclusion of objective 2, AllType FASTplex version 2 provided 100% concordance with version 1. It solved exon 1 ambiguity in HLA class 2, and now you can do single tube library processing. And finally comes the objective 3 of this presentation, which is validating the alternative protocol for FASTplex version 2. And I would like to share with you the timeline or the protocol that we use in our lab. This is our lab protocol, and it's not what One Lambda recommended. So for instance, we do in day one, which is usually start in the afternoon, we do the long range PCR and then we leave it overnight. And in the next day, we do the purification, then the quantification, dilute, barcoding, and then pooling. And then in day 3, we do the quantifying and then the sequence. The reason we couldn't do combine day 2 and day 3 at the same time because this is almost like a 7 hours protocol, and by the time the day end, the technologies become exhausted, and this step has a little bit of calculation, so we decided to move it to be done fresh in the next morning. In day 4, the TSV start to analyzing and then exporting the result to histotrack, and by day 5 we do the reporting. So in collaboration with the One Lambda, we decided in validating an alternative protocol, and the idea of this alternative protocol is that we decided to eliminate the purification step after day 1. And this actually in our hand takes almost 1 hour. And by eliminating this purification step, we were able to combine day 2 and day 3 into 1 day, which is almost 6 hours. And instead of reporting by day 5, we were able to report by day 4. So One Lambda recommended in order to proceed with this alternative protocol is to use a very high quality DNA. So this is a picture of a tap station showing the DNA that we used in this study. And you can see that all the DNA was, you can see a very good band around 60 kb. Band number 6, there was is a little bit faint, and this was a buccal swab because we accepted the challenge to add a buccal swab to this assay. And after we did our long range PCR using the alternative protocol compared to the recommended protocol, we also run a tap station. And as you can see here in the middle is the tap station using the recommended protocol, and you can see there are 3 extra bands in the middle which represent the exon 1 for DR, DQ, and DP. The top band is the 11 loci band, and the lower band represent the primer dimer. If you look at the bottom tap station for the unpurified amplicon using the alternative protocol, you can see that we still have the same trend of the 3 band that represent exon 1, the 11 loci band, but you can see that the primer dimer were a little bit has a bigger chunk at the bottom of the bioanalyzer because this sample was not purified. Interestingly, comparing the AllType FASTplex version 2 using the alternative protocol with the AllType FASTplex version 2 using the recommended protocol, we were able to achieve 100% concordance in the 24 sample that we analyzed. So we noticed that 3 sample there was high background in the alternative protocol compared to the recommended protocol. So A25 there was almost 20% false calls, but you have 542 depth of read sufficient to give you the right call. So we still have the ability to call it A25. The DQ alpha 01, we also notice false calls of C slightly above 20%, but you have 192 depth of read for the G, so we can still call the G as the right call. But with the DQ alpha 05, the chance was minimal. It was 50-50. So the software called this as a novel allele. However, we didn't call it as a novel allele because the number of reads were sufficient for that reason. The other thing that we notice with the alternative protocol is there is a slight low exon 6 coverage, especially with the DQ beta O3. And because of that, we were not able to rule out the DQ beta O3 454. And as you can see in this figure, you can see that the coverage in exon 6 is slightly lower than the other. In the end of conclusion of objective 3, AllType FASTplex version 2 using the alternative protocol provided 100% concordance with the recommended protocol. It's a very promising tool to reduce the hands-on time, and still a little bit of work needed to complete this alternative protocol. At the end, I would like to thank the histocompatibility and immunogenetic staff at the University of Saskatchewan and St. Paul Hospital. And I would like also to thank One Lambda for giving this opportunity to me to discuss this 3 different projects. I'll be happy to answer all your questions.